So far, we have regarded our book of life as a single manuscript that we all share in common. But because we have two sets of chromosomes, one from each parent, we actually carry two slightly different versions of it. And each of these versions is slightly different from everyone else's, except in the case of identical twins. It is these small differences in DNA that set us apart as individuals.
Our visual system is very good at recognizing subtle differences in facial features, for example. Chimpanzees probably think we all look alike, but these differences all go back to small differences in the genetic code.
So a full understanding of our DNA will also require knowing how it varies between two matching chromosomes, and then between any two individuals on Earth, and then learning what effect all this genetic variability has on our biology.
It's genetic variability that makes some of us tall and some short. It's genetic variability that makes some fat and some skinny, and that makes some predisposed to diseases like cancer.
What inspired the Human Genome Project in the first place was our desire to find those DNA variations that underlie human disease. Some genetic diseases are caused by a mutation in a single gene. Cystic fibrosis, for example, but these are the exception.
Most diseases and complex conditions, even aging, are not the product of a single broken gene, but of the interplay of many, many genes. These are called complex traits. They also become susceptible to analysis if you have all the genes at your disposal, because you can start to see which ones are varying or interacting in particular individuals or families to produce a particular disease state.
In fact, almost all the chronic ailments that plague us are due to the combined effect of a number of gene variations, which in turn interact with lifestyle and environmental factors. But these groups of genes are far more difficult to track down than the single mutations that cause simple genetic diseases, like cystic fibrosis.